Thank you so much for inviting me to speak about our project. So the project that we got funded from the KSF is a collaboration between my group, myself and Agnes and Carl Broman in Wisconsin. And we're facing this question that we've had for a long time. And some of you may have thought about this question is, why is it that some patients don't get parts of the phenotype? So if you look at this table with just the percentages, if you look at the congenital heart defect, there's only about half of the people with Kabuki syndrome that end up with a congenital heart defect. It's similar, maybe 40% for cleft palate and so forth. And there's even been in the literature cases where people felt like even they only had a very small subset of the phenotype, maybe only the congenital heart disease, but not much more. So what could explain this? And we see the same effect in mouse models. When we've been working with our mice and breeding them to different strains, we've seen shifts in the severity of the phenotypes. And one way to explain this observation is that there are other genes, so-called modifier genes, that kind of affect the eventual outcome. And so you can map these modifiers. It's a lot of work and needs a lot of mice, but that's the project that we have ongoing. And we have picked about 6 phenotypes. I'm showing you 5 of them where it's easy to measure them. It's IgA levels, it's an immune phenotype, the body weight, the number of kidneys, hearing, and then just the survival of the model. And all of these can be done relatively quickly. And then we're going to use a big resource in mouse genetics, something that's called the collaborative cross. And people have made these isogenic, or they're like monozygotic twins. They always have the same genetic variation on both alleles. And each of these mice have been studied quite a lot. And we can then, the first phase of this grant was to put the Kabuki mutation on each of these founder strains. And when we did that, what we see is similar to what I was describing before, that for some of the phenotypes, what I'm showing here is all the different founder strains, and then the phenotypes that we're measuring. And I'm showing you that for some of them, there is no difference between the wild type littermates and the ones that carry the Kabuki mutation. So in that strain, there's some genetic variation that protects against the Kabuki mutation. And you can see it's quite insightful when you look at it, is that any one strain, although this one is pretty good, but no one strain can rescue all the phenotypes, but we do see rescue of each of these phenotypes in some strain. So that means that we can, there's a really good chance that we could do mapping. And the way we, just to show you this data a little bit further, what you can see is that when we compare, I had shown you there's a big difference between the mice that have the Kabuki change versus the wild type littermates. But in these strains that I was showing you with the green, there's no longer any difference in the measurements. This is, for instance, weight, which is pretty interesting, easy to understand. You can see that the curves, the red curve, which mice that have the Kabuki variant, and the blue curve, which doesn't have the variant, there's no difference there. So there's been rescue of this growth phenotype, and you can see the same for the hearing. The mice had a hearing loss on the black 6 background, but it goes away in this NZO background. Okay, so once we figured that out, what the community has done is that they have taken all these strains and they've bred them many times together to create 2 kind of banked strain banks. So the thing that we're going to use is something called the collaborative cross, where they know exactly which piece of the chromosome the mouse got from each strain. So then we can breed the Kabuki mutation onto all these different mice. And there's quite a lot of these. There's about at least 50, maybe 60 such strains. And then we can look at the phenotypes and ask, when does the phenotype rescue and when does it not rescue? And then we can go to our collaborator, Carl Broman, who's done quite a lot of this. And he can tell us the regions that contain the area that is protective for some of these traits. And this will be an unbiased way of getting new therapeutic targets for Kabuki, but because this will kind of help us understand the underlying mechanism, what is going wrong in Kabuki syndrome in a very unbiased way. So it's not dependent on what we know now. And we're actively doing this now in batches of 10. And I don't have a lot of data to share at this stage, but this next phase is going to be a lengthy phase where we just systematically go through as many strains as we can, look at the phenotypes, and then the data will be shared with Carl, with the hope that we can find these little chromosomal segments that maybe have 50 or 100 genes or something that we can then evaluate in our assays to see if they carry a genetic change that could be useful to know about, that could infer new pathways to target and so forth. So that's all I wanted to say. I just want to thank my funders. I've been funded by the Luma G Foundation by a generous grant, and the Kabuki Syndrome Foundation, the Benjamin family and other families for this research. And these are all the people that work with me. So thank you so much. And I'll take questions at the question and answer session later today. So thank you.